Welcome you all to this cryo-EM course. So, today we are going to discuss about the sample preparation. Previously we discussed about the microscope anatomy, and we also know that near the goniometer or stage we are inserting our samples. There we are using the TEM holder, or there is a specific types of autoloader system that we are using for cryo grid insertion. Now, what is that grid, or if we just insert that grid, that is all for imaging, or do we really need to prepare our samples? So, initially when you started we discussed that this course will be mainly for biology students. So, then definitely our sample will be biological samples. So, those samples will be biological macromolecules. It may be only protein sample, or it may be DNA protein complex, it may be RNA protein complex, or it may be protein which is interacting with the liposome, or there may be samples like bacteria, viruses, or different types of cells like neutrophil or macrophages or erythrocyte, or we want to see organelles inside the cell. Those are all our samples, and we have to know how to prepare those samples, and it is a vast area, that sample preparation, and that will be one of our major topic for this course, how we should prepare our sample. And then we will discuss about when we will prepare our sample, how do we image our sample, and how do we using this electron dose to image, where you will get a good contrast images, and from there we have to decide that okay, which images are bad images, which images are good images, that we have to select, and we have to know how do we apply some kind of criteria to select the good images, bad images. And here we have to remember one more thing, that okay, there are two different types of samples we can consider. One is a group of researchers only use TEM or cryo-EM or SEM to take some snapshot of their samples. That sample may be bacterial sample, or there may be some kind of between the bacteria there will be some pili or some nanotubes, or some bacteria has flagella, after mutation or knockout some gene flagellas are disappeared. In those cases you just want a snapshot of that particular bacteria, or knockout strain of that bacteria or cell, or maybe you treated with some drug and then there are some morphology changes. So, you should have a control cell, and then after drug treatment whatever morphology changes happened, you want to image those things using TEM and SEM. Nowadays cryo is there. So, in cryo condition you can image something in a physiological condition, and those cases what we are doing, we are just trying to use our sample at cryo condition. So, it will be much more physiological condition. So, one set of researchers are going to use TEM, SEM or cryo-EM to just get some snapshot of their samples. On the other hand, we use this TEM or cryo-EM for structural study. So, we want to know the morphology of the cell or morphology of a bacteria inside the cell. We want to see how this lipid bilayer is forming inside the cell, or within that lipid bilayer is there any receptors are there, is there any channel is there. We want to see those things, and we want to understand what is the structure of that channel or that receptor or of that secretion system. Bacteria has a secretion system, we want to visualize those things and get the overall structure of that channel, transporter, or some other biological macromolecules whatever we have inside the bacterial cell or mammalian cell or any different types of cell. We want to characterize their structure, and those cases we want a structure at atomic resolution. Why we are talking about atomic or near atomic resolution? Because there we want to see that protein protein interaction means amino acid interaction, or any drug is interacting with any protein sample or a DNA samples or a RNA samples. We want to understand the chemistry behind that interaction, how that protein or RNA or DNA is interacting with one ligand or one drug molecule. Those cases we need a very high resolution data, and previously we are using negative staining microscope, means negative staining transmission electron microscope for structure determination, and we are generating a very low resolution model where we dock the atomic structure what we are getting from the PDB or using the X-ray crystallography or NMR. Nowadays EM lenses are very stable, detectors are superior, we have energy filter, our electron beams are very stable, which is giving us a very high resolution images, and that is the reason cryo-electron microscope nowadays we are using for determining high resolution structure of different types of biological macromolecules. It is starting from bacteria or cell, their organelles, to a very small protein, 100 kilodalton protein or DNA protein complexes. That is the reason it is a very wide range, and that is the reason it is better to understand the sample preparation. So, here I will give you couple of examples where we will discuss about the majorly negative staining TEM sample preparation as well as cryo sample preparation. So, same time we will discuss little bit about how we can prepare the samples for the cell or bacteria or viruses, as well as we will discuss about how we can get our sample which has a very small size like protein sample, DNA samples or RNA samples, and then we want to image and we want to get a very high resolution structure from those data. So, there will be a two different part, one part it will be focused on cryo, one part it will focus on negative staining TEM. Today I am going to discuss about negative staining TEM sample preparation. There at the beginning we will discuss about the protein sample preparation or DNA protein complex preparation, RNA protein sample preparation. Then how do we make those grids? For those grids we are going to use it in TEM, and we are going to insert those grid inside that TEM for imaging. So, we will discuss about those parts. Then we will go for sectioning using ultramicrotome or cryo ultramicrotome, and then finally we will go to cryo sample preparation also. So, sample preparation I will just use the notepad which will help us. So, as I mentioned whatever verbally I told, so we will just go through that cycle once, that will really help us. So, for any kind of biological sample preparation, or before we want to do cryo-electron microscope or normal TEM, it will be better we should have a proper planning. So, we should have a proper planning. Proper planning means, here as I mentioned that the today's topic we are going to discuss. First we are going to discuss negative staining TEM, and major focus will be protein sample preparation. Okay. So, in this protein sample preparation, it may be something like protein DNA complex or protein RNA complex or protein lipid complex or only protein. So, in all these cases, one thing is common if you pay attention, that is the protein sample. So, we are constantly talking about the protein with a DNA or RNA or something. So, if we just consider that okay, there is a only 2 DNA double helix or only RNA, it is bit difficult task to image only DNA or RNA, and imaging is still okay, but getting the structure of only RNA or DNA is bit difficult. When it will form a complex with a DNA or RNA or lipid, it will be easier to get the structure. So, that is the reason proper planning means what I want to say, that is the construct design. So, when we have the construct design, then so that means most of the cases whoever is coming from this biology background, you guys will be familiar that okay, so there will be a segment of DNA that we are just selected, we put this one in the vector, and that is called the plasmid, and that we transfer into the cell and we go for a over expression. So, here we will go for expression and then some biochemical experiment. So, these are the preliminary step I will suggest that okay. So, first step will be in your laboratory. So, we have to do this experiments in our lab, where we have a proper planning, means you go through a proper plan, then you design a construct, you clone those genes in your required vector. When you have those proper cloning, you take those things and perform some kind of expression test. This expression test means you have to go use some kind of stimulator, so like IPTG, to stimulate and get the protein, and then you have to do some biochemical experiments. Here biochemical experiments means first you have to find out whatever gene you have selected, you are getting your protein. So, first make sure that you are getting that protein. When you will get that protein, then you have to go to the next step, that purification. Purification of your protein of interest. So, this is a very important part, because for our cases, if we have, when you are cloning some gene, so you have a DNA, required DNA, you put it in the plasmid, and then you put it in the vector, and then you transform those things, and then you are getting your protein. You just consider that when you did that expression test, you are getting your protein. You are getting your protein, but when you will rupture the cell membrane and then you are trying to take out those protein, when you rupture the cell membrane, you can do different types of way, that mostly you can use the sonication also to rupture the cell membrane. Then you will use the centrifuge, all the cell debris will precipitate, and if your protein is soluble protein, that protein will go to the supernatant. So, you have a tube. So, what will happen, all these cell debris will precipitate here, and in the solution your soluble protein will stay. Then you will take this soluble protein, but when you will take the soluble protein, when you rupture the cell, what will happen? There are many other proteins will come into this solution. So, how do you separate your required protein? So, there you are targeting maybe one particular protein that maybe has a DNA polymerase or RNA polymerase or some kind of metabolic enzyme. So, you have to specifically select that protein only from the solution. In this particular case, you should know a proper method from where you can isolate that protein properly. And in this particular stage, purification is important. So, we are going to discuss about this purification step. So, if you have a sample like protein sample you want to do that, okay, there is a RNA polymerase or DNA polymerase, you want to see it how it is interacting with the DNA, and then you did the cell ruptured and there is so many other proteins, and if you just take after this expression and you rupture the cell. So, after damage the cell, you take this protein of mixture. So, mixture, protein mixture, you are trying to take this protein mixture and trying to image something using the TEM or SCM or your cryo-EM or whatever. So, what you will get in all the cases, what you will get? You will get a very heterogeneous population which is not very good for TEM imaging or cryo imaging, because those are high resolution tool and you can see all the protein in your micrograph here. So, and you can see a different shape of those proteins, some is circular, some is elongated, some has a different shape. So, and then from here it will be very difficult to figure it out which one is your protein of interest if sample is heterogeneous. That is one of the major reason we have to go for purification, and purification has certain protocols. We will go through these protocols like affinity purification, ion exchange purification or size exclusion chromatography, that we will discuss more. But when we will purify our protein, first thing first we should consider that protein and do some biochemical experiment. Biochemical experiment means if your protein has some activity, that if it is a metabolic enzyme, it will interact with some kind of ligand. So, first do those experiment, that is it interacting with your ligand or not, if it is giving you the product or not. So, those activity test or that binding between DNA and protein or binding between RNA and protein, if your protein has affinity to DNA or if your protein has affinity to RNA. So, better to do those biochemical tests in the laboratory before going to TEM or cryo-EM, because sometime that TEM and cryo-EM cannot tell you specifically that it is really interacting with the DNA or it is really interacting with the protein. Sometime you can see, yeah, DNA protein, after DNA protein complex formation, your protein is binding to the DNA, but you can confirm those things when you will do the biochemical test. So, that is the reason it is important whenever you will get the protein, purify the protein and do rest of the biochemical experiment, that if there is any affinity. So, sometime we have to use ITC, we have to use SPR or MST, some kind of biochemical and biophysical experiment you have to show. Same time, whenever you purify a protein, first thing first you should take that protein and check it how homogeneous your protein is. If protein is not homogeneous, you should go back and redo this experiment again. Until you get a very homogeneous protein, it will be better to work in the laboratory and try to get a better sample before you try cryo-EM or TEM. From TEM or cryo-EM, it is bit difficult to tell from the 2D images that which one is your sample. if your sample is not pure. So, this is our first step for any sample preparation, that first we will take make a proper plan, you clone, you construct design, express the protein, first check the expression is there. Whenever you express your protein, then next start checking that okay, whatever protein you are getting, is it your protein or is there some contamination is there, or is your protein is interacting with some other component. First check those things. So, better to do Western blot or mass spec to confirm that is your protein. When you confirm that is your protein, then go for the next large scale purification and do rest of the biochemical experiment, that if your protein has some activity, confirm those activity, then take those sample for TEM study or cryo-EM study. Now, take a look this slide that will give you the exact same idea that how we can use our TEM data. So, here from beginning I am starting from here. So, this is my first step. In this first step, as I mentioned previously, proper planning is very important. So, that includes your construct design, expression and some kind of preliminary biochemical experiment that you want to do, some preliminary biochemical experiment. When you do all these biochemical experiment, then you go to the next step where you can go for a large scale purification. In this large scale purification, you will get a good amount of protein. So, generally to taking one snapshot of a protein sample or a bacterial sample or a cell sample, you do not need a very large culture, but for structural biology purposes definitely we need a good amount protein concentration. If you compare, when we are comparing that as a structural biology tool, if you consider that cryo-electron microscopy or the TEM. So, what should be our protein concentration, what we should use it. So, generally for X-ray crystallography and NMR we need a huge amount protein sample. So, generally 5 mg, 7 mg, 10 mg is the minimum concentration is required to get a protein crystal or for any kind of NMR study. But for the cryo-EM study, generally a small amount protein sample is good enough, but that small amount is, if you compare to X-ray crystallography and NMR, but if you still that concentration is high concentration. So, generally for cryo-electron microscopy or TEM, we need minimum 1 mg to 1.5 mg protein sample. We will come back to those and I will say that that also depends on the size of the protein and that will vary, but generally 0.5 to 1.5 mg or 2 mg is a good concentration for the cryo-EM study. Now, the next step is the purification. As I mentioned previously that some biophysical and biochemical analysis are very important to check the protein quality, concentration and the activity. When we have all these things in our hand, if we have a feeling that our protein is pure sample, then we can think about go to the next step. What is the next step? Next step, we use a negative staining TEM. Now, you guys are familiar with the architecture of the TEM. So, this is our normal TEM where this is the goniometer or where we can insert our sample and then visualize your sample. Most of the time you can get an idea about the concentration of your sample and how homogeneous your sample is, how homogeneously it is distributed. So, after doing all of this, if you feel that your sample is not looking very homogeneous, when you check by negative staining, then it will be better to go back to the laboratory again and do these experiments again, go through this cycle again. If your protein is well known and if it is protein is already well characterized, something like apoferritin kind of sample or beta galactoside sample. So, if you check by quickly by negative staining, you will get an idea, or any protein that you are already working in the laboratory and you know that what is the overall shape, size. So, then probably you do not have to go through this one of these step, you will figure it out yourself that okay, there is a problem in my preparation. So, quickly if you check your sample by negative staining, it will always give us a little bit more confidence that how good is our sample. If it is not, we will go through this cycle again. But if sample is good enough, so generally we are taking that sample for the next stage. So, next stage means it is in cryo-electron microscope. This one is inside the box, the entire column and whatever component I discussed with you guys. So, here it is a 200 kb Talos Artica. So, where we can collect the screen the cryo-EM grid and cryo-EM data collection, and when we will do that and then we will use this data and go for structural characterization. Lot of time we face this problem when we collect this data. Sometimes there is a preferred orientation, or due to some reason in solution when we are freezing our samples are coming and aggregate. So, those cases we cannot use those samples for structural study or even for the snapshot. That time we have to go back to this stage again and we have to go through this cycle again. So, from the cryo-EM images also we can tell that how good is our data. If our data is good, then we can follow this path. If not, if there is a aggregation or if there is some kind of other problem that some part of the protein is falling apart, or there is some part is not very properly distributed, or there is some preferred orientation, those cases we directly go to the planning stage again and start from here. Otherwise, we will use the different types of algorithm to process our data, and there are couple of data processing tools are available, that is called the Reliant, EMAN, CryoSpark system. We can use this to characterize a 3-dimensional structure. So, finally, if we combine all of this, what we will get from this one, we will get the structure, and from these two steps we will get the functional characterization also. So, at the end of the day, whatever you are doing in this particular step, those will be also useful. So, before going to TEM study or cryo-EM study, we should pay attention in this steps and get a better data, better biochemical, biophysical data, and then go for the imaging study. Now, here this is the same part, couple of more things we have to add here that we are going to discuss here. That okay, protein expression, purification and complex assembly that we already discussed. Sometime for specifically this part, for DNA protein, RNA protein, lipid protein complex, what we need? We need the pure DNA or RNA or lipid or liposome separately, and you have to purify protein separately. If we think that the DNA protein altogether we are going to purify, so sometime it is very difficult or do the functional characterization that DNA protein interaction or RNA protein interaction. So, better those cases you purify your protein, you purify your DNA and RNA, all prepare your lipids separately and then check the interaction, do the biochemical study and then go for the next step. So, here when we are talking about purification, generally we prefer the two-step purification that I already mentioned, that purification that we are going to discuss in the next class, that affinity purification, ion exchange purification and SSC purification. So, here sometime if we can purify our protein properly, only one purification step is enough, but most of the time we use two step purification, that either affinity purification then SEC purification. SEC purification means size exclusion chromatography or gel filtration chromatography, or we do ion exchange chromatography then SEC purification. Whenever we will do this purification, many times we will lose our protein. That is one of the reason we have to go for a large culture and then do this purification step, because in every purification step we will lose some amount of our required protein. So, we have to keep it in our mind. If we go two step purification, at the end our protein concentration will reduce. So, that is the reason large culture is always good to get a good amount protein for TEM and cryo-EM study. And then when you have this protein, we should do this study that interacting with the DNA protein or RNA protein and protein lipid complex. It will be always good to perform those complex formation and run the SEC one more time, where we can isolate only the DNA protein complex or RNA protein complex or lipid protein complex. Sometime we want to see the complex formation between protein and protein. So, something like that, we have some kind of antigen and antibody is interacting. So, if we just consider that some kind of spike protein or that viral protein which is interacting with the cell receptor. So, those kind of protein and we have antibody against that. So, we want to make a complex that antigen and antibody. So, whenever we will try to make the complex between antigen and antibody, we cannot get a 100% complex. Some will be unbound antigen and antibody. Those unbound antigen and antibody we have to remove from our sample. So, those cases we have to mix them together, we have to purify both the proteins separately, we have to mix them together and we have to run the SEC to clean that. Only we will take the complex for our next study. Now, we will go through the rest of the part. When we have this sample, if we look at the slide, you can see it that after this purification, next step that okay, we need some time the buffer exchange, because remember that whenever we will try to purify our protein, most of the time our protein will be very happy and it will not aggregate, it will not precipitate at high salt concentration, generally 500 millimolar, 700 millimolar. And if we have a glycerol, for that glycerol or any organic solvent is not good for cryo-EM study, and that is the reason it is better to remove the glycerol. And if you have a high salt concentration, try to remove the high salt concentration, try to keep the protein at 150 to 300 millimolar and no glycerol will be the best. So, dialysis will be one of the method to remove the excess salt and buffer. So, sometime this is not going to work for each and every protein. So, based on your laboratory practices, you have to change this protocol little bit. This is some common protein that okay, generally soluble protein 150 millimolar salt concentration, no glycerol, they will be good. So, the fresh sample you will prepare and you will go for the structural study. Now, next, when you have this one, as I mentioned, you perform the negative staining, TEM imaging, 2D classification, we will discuss about all of this, and then if everything is working okay, you will go for cryo-EM data collection using Talos Artica or Titan Krios. So, those are two microscope from the Thermo Fisher what we are using for data collection, as well as there is a cryo arm from Geol is also there, which you can use it for cryo-EM data collection. Finally, we will do the image processing and the 3D reconstruction. If everything is working fine, you will resolve the structure and you can demonstrate their function also from that structure. If there is a finally, you will go for the model building, which will give you a near atomic model structure. And if you cannot get a proper structure, if there is a problem, generally that will give us from those experiment or optimization step, you will get some idea and you should implement those ideas in those steps, and then you do your you know final study where it will be possible to get your 3-dimensional structure. So, this is the basic outline how you should design your experiment and how you should do those steps. And next class we are going to discuss about that affinity purification, ACC purification or ion exchange purification. And also someone can consider that, okay, so why we are discussing about all of those purification steps, because this is very important if you want to do TEM or cryo-EM study. Instrument is one part, but these biological science samples are very heterogeneous, and the preparation, how to prepare the samples, it is also very important. That is the reason it will be better to go through those steps. Thank you all for your attention.